Hi, my name is Gabrielle. I am a third-year honors student in psychology at Concordia. Today I'll be presenting the research I performed during my Concordia Undergraduate Student Research Award in summer 2020. I completed this award under the supervision of Dr. Shimon Amir from the Center for Research in Behavioral Neurobiology, the CSBN. In summer 2020, physical access to the university was restricted, and I was unable to perform the experiment that I had planned. So instead, I worked on a literature review, which I'll be presenting here today. The title of this review is Modulation of Alcohol-Seeking Behaviors and Sex-Based Differences by the Circadian Clock. As the name indicates, the focus of this review is on circadian rhythms, which are basically our body's natural clock. So all of the oscillations that we experience within a 24-hour period, such as sleep or hunger, these are all under the control of circadian rhythms. These rhythms are synchronized with changes in daylight. They are present in virtually all of our physiological systems and in virtually all organisms. Research on substance use and circadian rhythms has consistently demonstrated a bidirectional interaction between clock disruptions and propensity for abuse. The largest body of evidence pertaining to this relationship has focused on alcohol, and a significant portion of this literature has identified embedded sex differences, adding a layer of complexity to this pattern. Alcohol use disorder leads to all-encompassing physiological disturbances affecting virtually all of our organs. Alcohol addiction bears numerous health and socioeconomic consequences, but its pharmacology remains ill-defined, leading to inadequacies in treatment. As mentioned previously, the use of alcohol as a psychoactive substance is bidirectionally linked to disrupted circadian rhythmicity. Alcohol has been shown to modify circadian physiology by acting on the brain's central clock in a region called the suprachiasmatic nucleus, and by generating circadian gene expression in other brain regions as well. In addition to these alterations, alcoholism also aggravates the negative consequences of an already desynchronized clock. In turn, chronobiological disruptions, such as changes in photoperiod, for example, jet lag, or chronic insomnia, promote relapse in former alcoholics. At the molecular level, alterations in circadian gene expression modulate drug-seeking behavior and cause changes in responsiveness to psychoactive drugs and other substances, including alcohol. Alcohol is also engaged in a reciprocal relationship with disturbances in sleep maintenance and especially sleep onset, further suggesting that complications caused by the substance interact with the circadian clock. As alcohol impacts circadian activity, physiological outputs of the clock also affect ethanol intake. This interaction demonstrates that as the relationship strengthens, the consequences can only be exacerbated. Now, there are numerous avenues to be explored within this research topics, and I'll just name a few here. Studies on sleep and insomnia indicate that appropriate oscillation of melatonin secretion, which is our sleep hormone, is inhibited in individuals under the influence of alcohol, and that in turn, disturbed sleep is a major predictor of relapse, if not the most important factor. Studies on reward sensitivity recognize that nearly all components of dopaminergic pathways, which are involved in reward and drug responsiveness, so these pathways follow daily rhythms as well. And studies on circadian genes and drug abuse suggest that mutations and deletions in core clock genes, such as the PER family genes or clock genes, are associated with increased patterns of drug seeking. In terms of sex differences, we know that historically females have been widely excluded from the literature, mainly due to their proposed instability in behavior and physiology attributed to the estrous cycle. Despite their underrepresentation in the literature, numerous lines of evidence demonstrate that alcohol use affects females differently and more severely than males. Namely, females experience alcoholic cirrhosis and alcoholic hepatitis more often than males. They attain neurotoxicity states more quickly. They have higher susceptibility to cognitive impairments due to alcohol. They are at higher risk for cardiomyopathy and cancer, and are more sensitive to the rewarding properties of substances of abuse. The underlying contributors to females' unique set of alcohol-related consequences rely heavily on the estrous cycle, but it's also been suggested to arise from differences in metabolism, differences in fluid composition, or from differential sensitivity or reward pathways. So a lot of avenues have been explored, but it's important to note that the inclusion of females in this literature is still emerging, and the implications of each factor remains mostly elusive. The main takeaway of this research is that the clock's influence spans widely, and this is reflected in the effects of its various physiological outputs on alcohol-seeking behavior. In studying chronobiology's authority over sleep, over the reward system, or our metabolic activity, we get a deeper understanding of the magnitude and complexity with which the clock operates. Importantly, clock gene deletions, or even polymorphisms, are associated with increased alcohol seeking, suggesting that these genes have properties which guard humans and other organisms from the development of potentially destructive behaviors, such as substance seeking. Just as desynchronization is associated with increased alcohol seeking and aggravated consequences, regularity in circadian activities has reversed effects. And as such, the circadian clock should be included in potential avenues for treatment of addiction behaviors. So I hope you learned a thing or two, and thank you for listening.